Yeah, welcome to today's last lecture. A few words in advance. So if you ever consider going into modeling, be prepared to be the last one in the presentations. With 100%, you're the last. Another preliminary comment is I've made some last minute changes in this talk in order to come back to some of those things that were being said earlier today in the other presentations. I hope it doesn't come across too confusing. So let me briefly introduce myself for those that watched the video lecture afterwards and haven't heard my introduction yesterday. I'm Kriest from the GEOMAR Ocean Research Center in Kiel, Germany. I started out as a biologist or biological oceanographer, but I quite quickly turned into modeling and later on into global models. So I'm maybe a bit of a mixture. So this is about mostly global models and remineralisation or respiration. I use these two terms as synonyms in a way. And if you have a look at global models, you come across many different names for the biogeochemistry. I show here just a few. Can all be very confusing to look at those models, and I hope I can guide you a bit through how these models work and how they deal with the various time and space scales we've heard about before. So what do these global biogeochemical ocean models do? Well, this is a bit vague, but it's meant to show the ocean thermohaline circulation. Typically, we represent the ocean by a bunch of boxes. So we discretize the global ocean. And of course, the global ocean is not a cube, but we have some topography in these models. So some regions are shallower, others are deeper. That's essentially how we consider the global ocean in its spatial dimensions. And then there are fancy physics in those models that calculate mixing, the up and down welling, advection, and so on via more or less scary equations. This will not be about these equations here. It's just these equations describe the mixing or the kind of transport of traces from one box to the other, either in the vertical or in the horizontal. And these models also have boundary conditions. So think of heat fluxes at the sea surface, of riverine input of fresh water, for example, or some slip conditions or so at the seafloor. This is all physics. And we immediately see, in our dreams, we would have an infinitely fine resolution of kind of 10 kilometers if possible. But of course, we see that if we want to have a very fine resolution, we require many, many boxes. And therefore, we would have to carry out a lot of these computations, and the entire thing would become computationally very demanding. So there is a limit to the spatial resolution that we can afford. This was all about physics. And now let's dive a bit into biogeochemistry. What these global models typically do is within each of those boxes, they compute the biogeochemical interactions. So typically the biogeochemical system is represented by individual compartments or traces or components, so here represented as these little rectangles with fluxes going in between. So for example, here flux from nutrients to phytoplankton, from phytoplankton to zooplankton, and so on. This is computed, this little biogeochemical model in each of those spatially discretized boxes. And then ocean physics takes care of kind of moving things from here to there and from here to there and so on. So this is essentially how these global models work. Ocean physics transports the biogeochemical traces around. there's also some exchange at the boundaries. Think of the acid gas exchange, oxygen or carbon dioxide, and dynamical equations calculate the interactions between those individual traces in each box. And again, we see that the more biogeochemical traces you want to represent, the more individual green rectangles we have here, the more expensive the model would become, because physics would have to transport all of these little things around, and also we'd have to compute many more of these interactions. So again, there is a limit to the representation of the diversity of the biogeochemical system. Okay, so far that was for now all about the global ocean biogeochemistry. Let's become a bit more 1D and look at just one single profile of boxes in the ocean. So typically at the surface, we have things going on like climate production, zooplankton and grazing, digestion, and these all supply the mesopelagic with dissolved and particulate organic matter. And this may be either through the physical transport, think of DOM or DOC, whatever, this mixing, subduction, and so on. I should note that in this case, with physical transport, each organic matter component comes in its backpack with an associated supply of phosphate, oxygen, and so on. So the organics are then transported with all the dissolved inorganics. And what is so interesting, and what we like to talk about so very much, is the sinking of particulate organic matter, because this moves organic matter through the water column instead of with the water movements. So organic matter is supplied individually. And I think this may be one of the reasons why we are also very interested in the active sinking fluxes. Together with remineralisation, this sinking of particulate organic matter produces vertical gradients in nutrients, oxygen, carbon dioxide. I think this is one of the things that's important to keep on the back of our minds. All right, now let's become even a bit more biological and look at sinking in particular and remineralisation. So we have an ensemble of particles at the sea surface, and it then sinks down, and we have less and less organic particles. On the way downwards, they remineralize, release phosphate, for example, consume oxygen, and so on. Very straightforward. Global models, I mentioned this before, they are somehow limited in computation time. So they typically combine a beautiful and usually very diverse particle ensemble in one or two components. From a biological standpoint, it's embarrassing, but that's just the way it is. These one or two components, compartments, traces are defined by differential equations with constants. And here's just one example how it would look like in paper. So consider D being detritus, computed, for example, in terms of millimole phosphorus per cubic meter, and coming together with phosphate, for example. So this is the time rate of change of detritus, the local time rate of change, say in this depth level of detritus, and it's governed by its remineralisation rate and its sinking speed times the divergence with depth. So we consider a single box, something sinks in from above and sinks out to below. And typically these things, these rates, come in units of per day. This is something like would more be presented today, earlier today. This would be the decay rate that's observed divided by the concentration of the aggregates, for example. And of course, there are things like sinking speeds. When detritus decays, so the phosphorus in detritus is being released as phosphate, so we increase the phosphate component by the same amount. And then associated with it might be a change in oxygen, if we consider oxygen in our model. And then the production of detritus, the decay of detritus would require oxygen, because even if we calculate detritus in terms of phosphorus, we know there's carbon associated with it, there's nitrogen and amino acids associated with it, so we require some amount of oxygen to respire everything to carbon dioxide, for example. So we also account for this here in oxygen by multiplying this decay with a stoichiometric constant. It's as simple as that. And so I put up this slide to make very clear what we require for the models and what the models will give us. So what the models require is, it requires ideas about the key players. So for example, here we assume we need detritus, phosphate, and oxygen. Very simple model. Could also think of other things, nitrogen, nitrate, etc. And then it requires things like these rate constant, like the remineralisation rate. These are typically being set beforehand. So for example, this could be something like 0.12 as presented by Morten earlier today. Or the sinking speed could be 10 meters per day. So these have to be defined beforehand before we run the model. And of course, we need some ideas about the equations. So what type of equation do we want to put in? Do we want to put in a Q10 rule or a Arrhenius rule to modify the remineralisation rate? And we usually get this information from people from the observing side, from lab experiments or in situ experiments, or sometimes just from gut feeling. What the model then provides us is, it steps this time rate of change forward in time, and it does so globally, because we are talking about global models. So at every point in the ocean, it will time step this equation forward in time, day 1, day 2, year 1, year 2, and so on. And advection of the physical part will take care of also moving on little detritus around. So in the end, what we get from the model are concentrations of our key players or components, detritus, phosphate, oxygen, everywhere in the ocean, at any time that was covered in a model situation, say from 1990 to 2100. This is the advantage of the model. We can look into the future in some way. And what the model provides us also, and that's another useful thing, is mass fluxes between the components over time. So for example, how much detritus is decaying and being moved to phosphate consuming oxygen over a year worldwide. In the model, we have perfect data coverage. This is also a very useful thing to have, because we usually only have scattered observations, but a global model provides us information everywhere in the ocean. And the interesting thing now is, what are the corresponding observations that we could use from the field to compare our model to see whether our model is indeed doing a good job, a reasonable job, because we want to see, put in all kinds of ideas into our model. We don't know whether it's right or wrong unless we compare with observations. That was a brief detour in how these models work, what they require, what they can provide. We don't always need a global model or a local one or a numerical one or a computer, but we can do many things beforehand on a sheet of paper and using a pencil. And here's just one very quick example. So we could assume, like we do here, we have n particles at the sea surface, and they all have the same mass and the same properties. They are all the same. And especially each particle sinks with the same sinking speed, say 1 meter per day, and it decays with the same rate to carbon dioxide, phosphate, and so on, say 0.12 per day. So again, we have a little time derivative of the detritus mass, and we can then assume that in the long-term equilibrium case, this term on the left-hand side is 0, so there's no change. It's comparable to a hemostat. And in that case, we can simply solve the right-hand side here analytically over depth. And we get a vertical detritus profile that says at a given depth that the detritus concentration will equal the detritus concentration at some upper depth, say 200 meters, times an exponential function of the decay rate divided by the sinking speed, multiplied by the distance traveled by detritus. So that's under the assumption that all particles sink with a constant sinking speed and remineralize at the same rate. And it can be solved just on paper. We don't need a global model for this, and not even a local one. Because they all sink with the same speed, we can do so analytically for the vertical flux, and we obtain a vertical flux profile that's an exponential function that depends on the remineralization rate. That's for those that don't see it very well. So that's here in the upper, in the numerator, divided by the sinking speed in the denominator, and then multiplied by the distance traveled. That's an exponential flux function profile. And we see on what kind of assumptions this depends on. If you plotted the flux here on a log scale, this is the green line over depth. You see, of course, on a log scale, this would be a straight line. So that was a very simple case. And of course, we know the world is not like that. But there are many factors that influence the particle sinking, such as size. Just think of Stokes' law, or old observational things from Smeda related to phytoplankton cells and from Alice Aldrich. Ballast might make things heavier, or zooplankton vertical migration might impact the particle flux in a way. Temperature via viscosity and Stokes' law might affect the particle sinking speed as well. And then on the other side of the coin, and the other side of the coin in this case is really remineralization rate divided by sinking speed. On the other side of the coin are factors that influence remineralization. So I could think of, again, size via the surface to volume ratio, the ballast of shells, or shells might protect organic matter from decay. Age of particles might make them less delicious. So think of some cold pizza pieces from yesterday, so nobody wants it. Giorgio, except Giorgio. Temperature, we've heard a lot about the Q10 rule and the Arrhenius rule. I won't dive into that here. And of course, oxygen. If there's no oxygen, the bacteria can't respire. Are these considered in global models? I would say yes, to a certain extent. Indeed, global modelers have been interested in all those different processes. So for example, the effect of aggregate size and ballast was considered in the model study by Mario and Guillen in 2006 in the French global model. Also, Jörg Schwinger from Norway looked at aggregates at a global scale. Zooplankton vertical migration was embedded in some models. The viscosity effect is in one Earth system model that's participating in CMIP 6. And likewise for remineralization, ballast is considered in some models. For example, in the UK Earth system model, Olivier Aumont looked at age. Many models considered either the Q10 or the Arrhenius rule. And also many models parameterize oxygen-dependent remineralization. So indeed, there are individual model studies that like to look at those things. And in case you're interested in the details of those parameterizations, at the end of this talk, the PDF I will provide, there is a list of literature with hyperlinks, and you can just look at those things. We were talking about size, and Morten has shown how these very beautiful and interesting things about aggregate. So let's stick with the on-paper calculation for a little bit longer. And let's consider the potential effects of size on particle flux. So assume we have a size distribution of particles at the sea surface. An ensemble of n particles, they all have a different size-dependent mass m, and each particle sinks with its own size-dependent sinking speed. For example, according to the empirical parameterizations we get from aggregate observations. So the full particle ensemble would result in the detritus mass that is pretty straightforward. It's simply the sum over all number of particles times the individual mass, and likewise the sinking flux of all particles would be the same, but additionally multiplied by the sinking speed. Very, very simple. Yeah, the sinking flux, for example, in this case, would look like this black line here. So very different to the exponential function. And now let's assume, as we've heard today, that each particle decays with the same size-independent rate r. And this relates back to these aggregate observations presented by Morgan earlier. And we can already imagine that the smallest ones, or the slowest ones, spend the longest time in the upper water column in particular, and will dissolve or remineralize first. And it will only be the large and fast ones that make it all the way down to the deep sea. So, if you look at this cartoon here, it's the small ones vanishing, and in the end, we end just up with the large particles. And if we were to compute an average sinking speed that is representative for the entire group, we immediately see that the average sinking speed, not the individual one, but the average sinking speed will increase with depth. In particular, if we would start from a size distribution at the sea surface that looks like this. So this is the diameter of particles on the log scale, and this is their mass on the log scale. So we start with a straight size distribution, kind of pretty common, I think. And then we go downwards here over these different lines. We see that's only the large particles eventually that show up at 4,000 meters depth. And as I said, the resulting particle flux would be this. And if we calculated the average particle sinking speed here, the flux divided by the concentration, we'd end up with this black line here. So somehow vertically increasing. This is again just an exercise that can be carried out on a sheet of paper and using a pencil. The unfortunate thing is that the solution of such an equation, starting with the straight particle size distribution and then propagating downwards, is a rather nasty function. It's an incomplete gamma function, depending on many things. And this can only be solved numerically again, so things start getting quite complicated. And also, we could go for many different size classes of particles, but as I told you before, the models can't afford this computation. So people really usually didn't go for this, but they went for the Martin curve. This is cheap. Just says flux at any depth z is proportional to the depth raised to some magic number minus b. This is our Martin curve, and it's empirically derived from these segment traps. So was it fit? The flux profile would look like this red line shown here. But it's just empirical. So far, it doesn't have any a priori meaning. But again, we can use a sheet of paper and a pencil and carry out some calculations, not too complicated, and we can show that in equilibrium, in the long term, if we assume a constant remineralization rate, but a sinking speed that increases vertically with depth, just as shown here, in equilibrium, we would end up with a margin curve with B given by remineralization rate, divided this time by the rate of vertical increase of sinking speed with depth. This is just straightforward math. We could also make some slightly different assumptions, assume that the remineralization rate decreases with the inverse of depth, and the sinking speed is constant again. We would end up with a margin curve and with some meaning for B. And this is very handy because now in hindcast, we can interpret changes in margin B by, for example, changes in the food web dynamics, such as small zooplankton with small fecal pellets and large zooplankton salps with large fecal pellets. We could think about the formation of aggregates affecting the sinking speed, or the oxygen availability affecting the remineralization rate, or temperature affecting either the remineralization rate via Q10 or Arrhenius, or the sinking speed via the viscosity of seawater. Why do I do all this? So first to show you that, well, we don't always have to go for a numerical model, but also because I try now to sort out what the models do in a kind of coherent way. And just consider we divide the models into two classes. Some models that apply a margin curve, also a direct flux function, or models that simulate detritus explicitly, detritus in this case described by a sinking speed and a remineralization rate, and then have a look at what global models do. So this column here are the models that go for a flux function, the other ones that consider explicit detritus. And these different rows show us the assumptions, for example, about the sinking speeds that are inside those models. And we see that some models assume a sinking speed or an exponential flux function, and some a margin function in a way, or vertically increasing sinking speed. Very few models assume a flux function based on size distribution. Some models resolve the size spectrum in a more or less realistic way. But we also see that with the assumptions of these flux functions, the models are basically all over the place. And as I mentioned before, sinking and remineralization, just two sides of the coin. The situation is quite similar for the parameterization of remineralization in those models. Most models assume temperature dependency or oxygen dependency, but then they are all over the place. So that's the summary of global models. More details, by the way, are in particular whether models include dissolved organic matter or not, are in this REMO recording from the REMO seminar series, which I strongly kind of recommend watching because these are really very nice. So most models include at least one explicit detritus component. Not all models include dissolved organic matter. And the key characteristics of those global model parameterizations are either a constant sinking speed or vertically increasing sinking speed usually. And the remineralization of organic matter itself is very diverse and depends on many things. I don't want to cover the full diversity of model simulations and model types here because this is a bit in the making here also during this project. And this is an impressive kind of challenging task. One thing to look at would be the paper by Stefan Johansen that was mentioned by Carol today. But let's have a little bit of a look for the end, for the next few slides, into investigating some consequences of certain assumptions about sinking remineralization in the model that we also going to use in the lecture. So that model is a pretty simple biogeochemistry. It contains, in short, it contains 3 elements, phosphorus, nitrogen, and oxygen. It assumes a fixed stoichiometry in the organic components. So the biology is represented by phytoplankton, zooplankton, detritus, phosphate, and dissolved organic matter. And there are processes such as denitrification, nitrogen fixation, and oxygen included in that model. The model is run in several global circulations, and in the following, I'd like to focus on some simulations I carried out with the TMM, the transport matrix method. This is an offline circulation that repeats the same circulation mixing every year. And it's very efficient. I can run this over long times and carry out many experiments with all the uncertain rate constants that go into the model. Just very briefly on the details, the model assumes a vertically increasing sinking speed of detritus, as we had before. And for the remineralization of detritus and dissolved organic matter, it assumes that the remineralization rate depends on a saturation curve of oxygen. So as oxygen declines, the rate will go down. But it also assumes a second oxidant, and that is nitrate. Some bacteria can switch to denitrification, the implicit bacteria. And in that case, denitrification is limited by the oxygen availability and is also represented by a saturation curve of nitrate saturation. So 2 ways to inspire particles. And in that model, again, coming back to our paper analysis, if there's no oxygen limitation, this one term, well, this term will become 1. There's sufficient oxygen, and we would end up with a Martin curve in the absence under sufficient oxygen concentrations. So, as I said before, this is very efficient. I can do very long-term simulations with this global model, and I can also play around with the Martin V, for example, and this is what I did. So what you see here is the global average oxygen concentration expressed as difference from where the model started. I start usually at World Ocean Atlas data set distribution. So a 0 means the model has exactly the same amount of oxygen as the World Ocean Atlas. And on the x-axis, you see the time as I run the model forward over 9,000 years. And on a log scale. And remember, this circulation isn't changing over time. It's the same every year and every year. There's no climate change in that model. It's just boring. The 3 different lines here indicate now different margin curves of sinking speed assumptions are plugged into the model. And the blue one is a rather high Martin V of around 1, representing rather slowly sinking particles. And the red one is a rather low Martin V, representative of a very fast increase of sinking speed with depth, so rather fast particles. So what you see here is something very interesting. So initially, oxygen goes a bit up in the ocean. The first year, the ocean is taking a bit of a breath because the water might be a bit cold and there's a lot of uptake through acid gas exchange. But then oxygen starts to decline in the ocean over the next 100 years. So this is the acid gas exchange, this initial increase, and this is happening very quickly. It's quite big, a quite vigorous process. But then remineralization kicks in and respires the oxygen in the ocean. And how strongly this occurs depends on the Martin V we put into the model. Now, eventually, for the slow sinking model, this decline in oxygen recovers again, whereas it continues for the fast sinking model. And here we now see the combined effects of this remineralization in the deeper ocean and circulation, because with slow sinking, we will have shallow remineralization and a lot of oxygen will be used up at the surface on the mesopelagic, and see the ocean surface and the atmosphere quite quickly again. So eventually, the entire ocean with its biology chemistry, if we consider it as one system, just such that there is some respiration going on in the mesopelagic, but it is quickly refueled from the atmosphere. Whereas if we have fast sinking, a lot of this depth, a lot of this demand on oxygen will be in the deep ocean, and this takes ages to see the and long time scales to see the atmosphere again. So eventually the entire model ocean adjusts such that this model ends up with a large oxygen deficit. So here we see really the combined effects, not only of biology chemistry, but also of large scale circulation and all the very long time scales and the global scales. If you want to have a little bit of a different view, because it's so late, and I thought it would be good to also show a movie. This is here now, towards the end of this talk. So what you see here is observed oxygen in the upper 100 meters, and here between 2,000 and 6,000 meters. So this is from World Ocean Atlas. The color scale indicates oxygen, so dark brown is high oxygen, dark blue is low oxygen. And on the right side, you see the model. So it's a bit difficult to see here, but in this case, I'll start the model with 177 millimole oxygen everywhere in the ocean. So it's just a uniform color. This is year 0. This is from where the model starts. And now I will run the model forward in time, and what you will see is a quick adjustment of the surface with regard to oxygen, and have a look at the areas of deep ventilation here in the North Atlantic and the Weddell Sea, what's happening over time. So here above, you will see the year as the model runs forward in time, 100-year time steps. So the ocean at the surface is taking very quick breath, and now we see what's going on in the model here in the deep ventilation areas. Here, oxygen has declined a bit. That was a bunch of particles sinking down, being respired, but now it's being refueled again from the Southern Ocean, and slowly oxygen is propagating downwards. I really apologize for the colors. These should be blue. So you see, to some extent, regardless from where the model starts, we use the circulation, we use the biology chemistry, we run the model forward in time, and if we are lucky, the model ends up with distributions that at least to some extent resemble the distributions that we have in the real ocean. And it happens quite quickly at the surface, but it takes quite some millennia to happen in the deep ocean. Just to explain why this is happening, so consider ocean circulation here, or global ocean overturning here. This is the Southern Ocean with the Antarctic circumpolar current going round and round. This is here the Atlantic, with the North Atlantic deep water formation and Arctic bottom water flowing back to the Southern Ocean. On the right-hand side, you see the Pacific with no deep water formation areas. And what we put into this is how the propagation of water masses in this case works. And now we put on top of that, or particles sinking, for example, fast or slow, and decay on the way. And we see that at the global scale, how the interaction between biology, chemistry, and large-scale circulation will eventually affect the global distribution of traces, but also, in case of oxygen, for example, may affect the inventory of oxygen in our model ocean. So let's come back to a little example. We've seen this plot here with the 3 different lines over time. And I said I can play around quite happily with the parameters because the model is quite efficient. So I can also play around with the remineralization rate constants, so for example, the nitrate affinity or the oxygen and nitrate affinity for remineralization or respiration. And have a look now at 9 different model experiments. Have a look at how well the model matches the global inventory. Remember, the 0 line would tell us the model has a good global inventory of oxygen and possibly could use this inventory as a measure to judge how realistic my model might be given this particular circulation. What we see is overall playing around with the oxidant affinities doesn't seem to have a very large effect. These 3 different panels, they look pretty similar, right? But the B seems to have a considerable effect on the oxygen inventory. So we could use, via this long combined circulation biology chemistry effects, use things like the oxygen inventory as a measure to tell us how well the model performs given certain parameterizations of remineralization. But this is somehow very far away from where we started from the real biology. All these fancy little particles and observations. As I said before, we can also look at the global fluxes of the model. So how much we try to sustain remineralized, or how much we try to sink through the water column. And this is finally shown here. So this color shade shows you the particle flux at some depth, I think 2000 meters. So red means a high particle flux, blue means a low particle flux. And these little dots show sediment track observations from the Honyo data set, I think. And then we can compare the models side by side as the model match observed particle flux. This is shown here. So this is the model flux, millimole carbon per square meter a year. This is the observed flux, both on a log scale. And these are the results here, these little red dots. It's a cloud in a way, but this is as good as it gets. It's not specific to this model. These things are usually very cloudy. Nevertheless, we can do fancy things, calculate a root mean squared error, a correlation, a bias, whatever, because we want to see which model and which model setup is the best. So we can do this for all our 9 and more model experiments. And this is finally shown here. So what you see here on the x-axis is the B in all or different model experiments, the different colors indicating different affinities for oxygen and nitrate, and the y-axis showing the root mean squared error of this analysis here for each of the individual model simulations. So overall, we find that the lowest root mean squared error, the best model, is something around a B that's about 1-ish. And again, we see that however we change these saturation constants of remineralization, doesn't seem to make a big difference. But it seems to be the particle flux curve that matters a lot. We could also look at the bias against this data set. 0 bias would be perfect. Again, we see B around 1, which in this case seems to be best. This brings me to the end. So this is the one side of the coin. This is particle flux. But we have the other side of the coin, which would be remineralization. Better, so flux from detritus to phosphate. This is still an open question. This is what REMO is about. Finding good constraints for model assessments to determine which model does a good job, which model doesn't, and also finding good rate constants, rate parameters, and parameterizations. So this is what this project is up to. With that, I'd like to finish this very long day.